The following method offers reliable and sensitive detection of specific chromatin modifications on selected plant genes. First, cross-link modified histones and DNA with formaldehyde. Then, extract and sonicate the chromatin to facilitate immunoprecipitation with modification-specific antibodies. Finally, analyze the ChIP reaction by the decross-linking of histone DNA complexes and gene-specific real-time quantitative PCR. In plants, this approach can provide molecular insights, like the specific histone modifications associated with C4 photosynthesis in maize, and systemic immunity in Arabidopsis.
The main advantage of chromatin immunoprecipitation over existing methods like mass spectrometry is that this technique allows the detection of histone modifications on selected sequences in the plant genome.
Generally, individuals new to this method will struggle because the protocol entails a couple of steps whose correct performance is key to success of the procedure.
In this video, we use ChIP analysis to provide insight into the regulation of gene expression in the model plant Arabidopsis thaliana. Demonstrating the procedure will be Michal Jaskiewicz, a PhD student.
For each sample, begin with a harvest of 2 grams of Arabidopsis leaves in a 50 milliliter plastic tube. Fill to the 40 milliliter mark with cross-linking buffer and position a tailored filter sponge right above the buffer surface to ensure the leaves stay immersed. Now place the samples in a desiccator with vacuum infiltrate for 10 minutes. To stop the cross-linking reaction, add 2.5 milliliters of 2 molar glycine and mix by inversion. Then infiltrate again for 5 minutes. Continue to vacuum infiltrate for another 5 minutes. Then discard the fluid while harvesting leaves on a sieve. After washing the leaves twice with 1 liter of water in a plastic beaker, dry the leaves thoroughly with paper towels. Collect leaves into a fresh plastic tube and freeze in liquid nitrogen. Store the samples at -80 degrees Celsius until chromatin isolation. Using a liquid nitrogen-cooled mortar and pestle, grind the frozen samples to a fine powder. Transfer the powder to a 50 milliliter plastic tube, suspend the powder in 30 milliliters of extraction buffer number 1, and incubate for 15 minutes at 4 degrees Celsius on an overhead shaker. Pass the suspension through 4 layers of mirror cloth into a fresh 50 milliliter plastic tube. After centrifugation, discard the supernatant. Next, use a paintbrush to resuspend the pellet in 50 microliters of extraction buffer number 2. Then add another 950 microliters of extraction buffer number 2. Transfer the suspension to a 1.5 milliliter microfuge tube and centrifuge for 10 minutes. Meanwhile, prepare 2 milliliter microfuge tubes containing 1.5 milliliters of extraction buffer number 3. Next, discard the supernatants. To gradually suspend the pellet in 300 microliters of extraction buffer number 3, first add a small volume of buffer number 3, suspend the pellet with a paintbrush, and then add the remaining buffer. Now carefully layer the samples on top of the prepared tubes of 1.5 milliliters of extraction buffer number 3, ensuring the 2 phases won't mix. Centrifuge samples for 1 hour at 16,000 times G at 4 degrees Celsius. Discard the supernatant and suspend the chromatin pellet with a paintbrush in 300 microliters of sonication buffer. Sonicate chromatin to a DNA size of approximately 400 base pairs. When sonicating, make sure chromatin will not be heated above approximately 30 degrees Celsius to protect heat-sensitive crosslinks. Centrifuge the sonicated sample to precipitate undissolved material and harvest the supernatant. To 2 milliliter microfuge tubes containing protein A agarose and antibody binding buffer, add 200 microliters of the sample chromatin preparation. Incubate the tubes for 1 hour on an overhead shaker. After centrifugation, harvest the sample supernatants and aliquot 30 microliters of protein A agarose to fresh 1.5 milliliter tubes. For determination of chromatin concentration of input material, reserve 40 microliters of the sonicated chromatin. Then add 400 microliters of the sonicated chromatin suspension to the 30 microliters of protein A agarose and an appropriate amount of modification-specific antibody. Incubate overnight on an overhead shaker at 4 degrees Celsius. Harvest beads by centrifugation for 2 minutes at 440 times G. Remove the supernatant and perform successive 10-minute washes of bead pellets with 900 microliters of the respective buffer on an overhead shaker. After the final wash, completely remove any remaining buffer. To free the DNA from the histones, add 400 microliters of D-cross-linking buffer to the beads and the 40 microliter input sample preserved earlier. Vortex for 5 minutes, centrifuge samples, and incubate at 65 degrees Celsius overnight. Now purify the DNA with a commercial DNA purification kit and perform conventional locus-specific real-time quantitative RT-PCR. In this example, expression of the Arabidopsis thaliana PR2 defense gene was induced by benzothiodiazole, a synthetic analog of the plant hormone salicylic acid. Results demonstrate that activation of PR2 expression is associated with an increase in the acetylation of lysine residues on histones H3 and H4 on the PR2 promoter.
While attempting this procedure, it's important to remember that addition of extraction buffer 1 to the leaf powder causes overpressure in the closed Falcon tube. Please remember to open the tube once in a while. Also remember not to remove paintbrush after suspension of pellets before addition of the next buffer.
After watching this video, you should have a good understanding of how to determine histone modifications in plant leaves via formaldehyde-based cross-linking of DNA and histones, isolation of chromatin, immunoprecipitation, and locus-specific DNA quantitation to answer key questions on the role of chromatin and its modifications in different fields of plant biology.
Take into consideration. It is important to disrupt chromatin during sonication to fragments of about 400 BP. Exhaustive sonication will destroy chromatin by disrupting DNA and histones, whereas insufficient sonication will leave long chromatin fragments. Sonication efficiency is best tested by collecting 20 μl chromatin from samples before and after sonication, decross-linking DNA and histones, isolating the DNA and determining the length of the sheared DNA by agarose gel electrophoresis. RNA-seq should be added to the samples before electrophoresis, because the chromatin preparation still contains large amounts of RNA at this stage of purification that might interfere with the visualization of fragmented DNA. The samples are useful for chromatin immunoprecipitation if DNA from non-sheared chromatin is longer than 10 kbp, whereas DNA from sheared chromatin forms a smear with maximum signal intensity around 400 bp of DNA. Make sure to exchange your formaldehyde stock frequently as the compound tends to polymerize with time. Formaldehyde solutions are stable only for approximately 1 month, and this period is hardly prolonged by methanol stabilization. It is advised to test different formaldehyde concentrations when setting up experimental conditions. It is important to infiltrate low amounts of leaf material in a large volume of buffer to avoid leaves sticking to each other. Leaf sticking often results in insufficient infiltration of formaldehyde. Too much plant material will block pores of the mirrorcloth tissue. The grinding efficiency considerably depends on using a mortar and pestle with the perfect fit. Thank you for watching. Subscribe if it is useful.